Today, our topic of discussion is somatic embryogenesis and its consequences in cereals. To talk about, we have certain contents, that is, introduction, somatic embryogenesis, the factors affecting somatic embryogenesis, the types involved, the role of somatic genesis, embryogenesis in terms of cereal development, and finally, the conclusion. Now, when we talk about in brief about somatic embryogenesis, we get an idea about it's a process by which the somatic cells differentiate into somatic embryos. That is the plant embryogenesis, where the process it produces an embryo from a zygote by some kind of asymmetric divisions. So thereby, there will be a differentiation of an undifferentiated cellular mass which have the property of general cellular totipotency, referred as a callus. So somatic embryogenesis has the following major advantages when we compare to the zygotic embryogenesis. That is, the process of somatic embryogenesis can be easily monitored. The environment and the developmental phases of somatic embryo formation, they can be controlled, and the large numbers of embryos can be easily obtained. So today, back about a brief history of time, talking about the history and development of somatic embryos. Basically, in 1958 to around 59, in that range, F.C. Stewart and Reynard independently reported the formation of the embryos by the somatic cells of Docus carota, that's a carrot. Somatic embryogenesis process was held, and since then, the somatic embryogenesis has been reported in more than 500 different species of dicots and monocot, respectively. So, henceforth, we got an idea about the embryos which are actually formed by the process of somatic embryogenesis are primarily embryoids. Now, to talk about another most important aspect, that the most major requirement for the embryo development is a physical and the chemical environmental conditions. And the various phytohormones, the growth hormone parameters, and other nutrients that's been used, where we call the somatic single cell origin of the somatic embryos, they are indipped or they're imbibed inside a magic bath of embryo sac. So henceforth, this in vitro embryo, which can be developed if we provide this nutritional conditions or nutritional environmental conditions, that's quite same that's been obtained in the magic bath. Now, it's a very interesting picture to note that, and very conventional picture also, where we get a distinct differences between the zygotic, the concept of zygotic, and versus the somatic embryos. So if you see that a somatic cell development starts slowly from somatic cell, which divides to vacuolated cells, and from one cell proper, development of a quadrant stage, octant, dermatogen formations, early globular torpedo stages, and therefore the formation of linear cotillary stages. But when we understand the various basics about how zygotic embryos are very distinct from the somatic embryos, we get an idea about that somatic embryogenesis is an inductive process, and embryogenesis, they can take place without the involvement of fertilizations or genetic fusion. Early divisions, they do not follow the sequence in order in somatic embryo development. Suspensor is absent in somatic embryos. Somatic embryos do not become dormant, and they lack the endosperm differentiation in somatic embryos. So you don't have any vascular connections as soever in somatic embryos. Unlike the zygotic embryos, somatic embryos often shows secondary embryogenesis for the development, for the unsynchronous development. So therefore, zygotic embryos are usually diploid, and also that the somatic embryos, they may be haploid, diploid, or even triploid. So therefore, zygotic and somatic embryos, they share similar growth ontogenesis, the tissue development, with typically passing through globular, heart, torpedo, and quaternary stages. So globular, scutellar, and coleoptillar stages, they are being called in the monocot. And for the others, we call as a globular, heart, torpedo, and for the quaternary stages. So henceforth, the somatic embryogenesis, they also occupy or accumulate a seed-specific storage reserves and also the proteins, which are very much characteristic of the seed storage proteins in the lesser amount. Now, another most interesting concept when we talk about are the factors that are employed. Now, factors are very interesting because the developmental process of the somatic embryogenesis actually starts off with 4 phases. We start with the phase 0, the phase 1, 2, and 3. So certain important parameters are important when we talk about the sources, the explant. Explant are the prior source material which helps in the induction process of somatic embryogenesis. They are very diverse and they are very responsive plant, such as they've been found in any part of the plant that actually induce this embryogenic culture development. So therefore, please remember, guys, that they're also at times being used for very recalcitrant, okay, resurrective recalcitrant plants such as cereals and legumes, in which the explants they varies even in between the species. Now, there are various types of explants that can be used for the somatic embryo implantations. The mature zygotic embryos, inflorescence, cell suspension cultures, petioles, protoplast, leaves, stems, and roots. So there are varied ways and means. As I promised that there are certain roles of plant growth regulators which plays a pivotal role. For example, the auxin, the 2,4-D. We all know that the best synthetic auxin used for development or induction process of somatic embryos. So continuous supply of embryogenic callus development or calli development to proliferate inside the medium without the appearance of the embryo is actually thrust upon by auxin. So the continuous supply of auxin induces an endogenous ethylene productions, which also suppresses the embryo development. So henceforth, we get an idea about the various ways and means, the cytokines which are responsible for the development of the globular stages from initial embryo. And we all know the commercial zeatin, yes, it's actually been a promotive when they're applied to the embryogenic cells in after 3 to 4 days of transfers and proliferation to the medium, whereas the benzaldehyde and kinetin, they do have certain inhibitory effect on the embryogenesis. So therefore, the ratio is very vital. High ratio of cytokinin upon or versus auxin induces the shoot formation and reverse ratio, which favors the rooting. Polyamines, yes, polyamines, they also plays an important role in vitro and in vivo development of somatic embryos. That's involved in cell growth, proliferations, and aging processes, which are very interactive in terms of negatively charged molecules, specifically the oligonucleotides and also the proteins. Of the 3 polyamines being studied, putrescine, spermidine, and spermine, they are very drastically important for the increased development of these somatic embryos. Genotypic effect of somatic embryogenesis, they occur for the purpose of regenerations and differentiations. Due to the variations of various endogenous levels of phytohormones, we can modulate the development of somatic embryos automatically. Even a short electric stimuli can also promote the differentiations of the organized shoot embryo by affecting the cellular polarity through changes in organizations and the microtubule development for asymmetric divisions. Now let's understand the various stages being deployed for induction process. Now the first as I said, that the stages of development of somatic embryos start with phase zero, where the cells become competent enough in the presence of auxin, that's 2,4-D, for the inductions of embryogenesis. Second is a phase one, where the competent cells have the ability to develop somatic embryos when auxin is removed from the medium. The second phase two, that's a rapid cell division occurs in cell cluster, leading to the formations of the pro-embryonic mass or globular cells, right? And the third, the phase three, where the plantlet develop from the globular embryos via heart-shaped or catenary phases. So therefore, you can see that unlike the seed embryo development, somatic embryos do not go through the final phases of embryogenesis, which is called the embryo maturation, which is characterized by the accumulation of certain seed storage or embryo-specific reserve food material and proteins, which impart desiccation tolerance to the embryos, but rather ABA, which prevent the precocious germinations and promote the normal development organogenesis by triggering the expressions of those genes, which normally are expressed during the desiccation phase or the drying down stages of the seeds. Now, let's understand what are direct somatic embryos. Direct somatic embryos are actually when the embryos are being formed from the explants, and they're quite identical clones without the interventions of the callus development. The explants, they're capable of direct embryogenesis development, and they seem to carry certain competent somatic cells in them. They are called the pre-embryonic determined cells, called the PET seeds. Now, these cells, they're committed, they're highly committed cells, destined cells for the embryo development and need only to be released. Whereas the indirect development of the somatic embryos aimed with the development of the mass of callus, which have natural capability of cellular differentiations or cellular totipotency, which is maintained or differentiated into embryos. So this redetermination phase of the embryogenic development pattern, which is actually called an induced embryogenic determined cells or IEDCs. So you can see the various stages of direct somatic embryogenesis, which starts with a fused globular stages embryo with an early heart-shaped structure, with an early torpedo stage, advanced torpedo stage, and finally, we have the catenary stages. So if you kindly see this panel of how the distinction have actually talked about recently, that how the distinct suspensor is absent in somatic embryos, where a well-developed distinct suspensor is present. So to add on, the embryos have no vascular connections for somatic embryos, and for zygotic one, they do have vascular connections with respect to their explant. So the question comes that uh, about the various advantage and disadvantages of somatic embryos. So when we talk about this particular large-scale propagations and the scope of the development, we get a varied sketch. Because somatic embryogenesis, there has been offered the opportunity to develop an efficient propagation system in large numbers of angiospermic plant. So micropropagation through the somatic embryogenesis has many advantages over conventional vegetative propagations, and it's very commercial use in horticultural applications and forestry and expanding worldwide. So it is observable that it's a various cultural conditions are being attuned, controlled with a high propagation rate, with a high somaclonal variations to preserve certain elite genotypic variations, conservation of germplasm, and elimination of diseases and viruses can be done by the usage of somatic embryos. But the true disadvantage lies when the somatic embryos shows a very poor germination rate because of their physiological, biochemical immaturities and instability of cultured cells in long-term cultures is a major limitations upon commercial exploitations as a mass propagation of towards somatic embryos development. So there are varied role played by the somatic embryogenesis, that's in terms of serial development, where the use of somatic variations comes into play, where the high frequency and the conservation of germplasm or elite genotype comes into play, or even the emphasis on recalcitrant seeds development have been formed. So therefore, one can state it in a very future way that so somatic embryogenesis would be a very important tool upon the conventional zygotic embryo development in terms of plant breeding, mass propagations, and rapid genetic improvement for the commercial important crops. So henceforth, several embryo-specific genes have been isolated from the embryogenic cultures, specifically when we talk about edible cereals and several molecular markers to distinguish between the embryogenic and non-embryogenic cultures, and definitely isolating these cells would actually trigger an advantage. With this note, hopefully, this wise owl presentations have benefited y'all guys. So we expect more subscriptions and more encouragement to see further videos. Thank you so much once again. And here, Dr. Ray signing out. Bye bye.